Welcome! Today we are going to do microscopes, but instead of just looking at, like, we're going to look at cells in the microscopes today. So the activity you picked up today has multiple different kind of parts to go through, and we're going to be observing organelles within the microscope. Now, depending upon the cells that we're looking at and the magnification that we have, will kind of depend upon what we can see. Right, so sometimes we can see some organelles in some cells, and sometimes we can see different organelles in others. So, we kind of choose what organelles we ask you to specifically look for when we are looking for each kind of cell. There's little circles throughout the lab that say, hey, draw what you see. Okay, we want to draw what you see. If you want, we could dig out some colored pencils for your pictures. They're on that back table, we could. I'm not super concerned about coloring pictures. We're more asking you to draw like the best of your ability, sketches of what you're observing. And even when we ask you to look at an onion, some onion skin, we aren't asking you to draw you don't have to draw like 20 onion cells. I think it just says draw a few. Okay, so you're not even going to draw everything you see. Clean up in aisle 3, Mr. DeVore. Yeah, literally 3. So, we're going to ask, so remember how we asked, we practice estimating sizes of things? We're going to ask you to do the same thing. We'll write it back up on the board again, our magnifications and our field of views. But if from here to here is 4.5 millimeters, we want you to approximate how many of those things can we fit across that 4.5. Then we take 4.5 divided by that number, and that will give us our approximate size. Okay? So we'll go through and we'll talk about that. We'll put that back up on the board, I should say, so you have that as a reference. And then we ask you to label some parts, and depending upon what we are looking at, depends upon what parts we're going to ask you to label.
With the drawing, do the best you can. You're not going to get a lower grade because, well, your nucleus sure looks funky, right? It's not art class. But try to be detailed. And a common mistake that we see every year, say draw an onion cell, right? If someone will draw like everything they see through their eyepiece. So this is one cell. This whole thing is actually, in this instance, is 5. So when it says draw one cell, find your, in the case of an onion cell, find your cell wall, cell membrane, and you just need to draw one. Okay, so here's one cell. There's my nucleus. Maybe you'll be able to identify some other components, but this is not, just because you see it through the eyepiece, that's not one cell. Does that make sense? We just need you to draw one. Oh, thank you. Yeah.
So again, if this is like our field of view, that was not a very good circle. But the idea is, again, estimating size, if from here to here, depending on whether we're on high or low power, is some known field of view. So if, let's say this is high power, from here to here is 0.45 millimeters. Well, then you're going to say, about how many of these things can I fit across? And however many that is, it could be 0.2, it could be 18, depending on how many it is. You take your field of view, divided by the number of objects you think you can fit across, and then that'll give you your approximate size. Okay, so I think for this onion cell, we're asking for length of the onion cell. There's a couple different other things. So onion cells first. The second thing is, we'll help you with the onion skin one. Purple plant, so we'll pull some of the leaves off of that, is Transcandia. I don't know how to pronounce the Latin wording of it. But we look at the leaves and we can just cut little slivers, because remember, the biggest we're looking at is 4.5 millimeters. So if you have a little piece of plant leaf that's like that big by that big, that's plenty. You wanna make sure you put the purple side up. There's some green on the leaf, but you wanna make sure like the all purple side is up. The Elodia is what's in that container on the Windows well right now. And we'll pull some of those. You're just gonna look at a single leaflet of that and we can help you with all that. All these you're making wet mounts. Let me just make sure that's clear. You're making a wet mount for everything. And a wet mount means slide, specimen, liquid, cover slip. The difference is on the onion cell, instead of using water for our wet mount, the onion skin is really translucent. So we're gonna stain it, and we're gonna stain it with iodine, and that's gonna be our liquid for our wet mount, because it will help stain. It's gonna stain, so be careful because.
Do you wanna do this, should we do the staining right now?
I don't know.
Just be careful, it's stained.
It's stained.
You don't want it on clothes.
It'll stain. Okay, it's stained. It's iodine that we're using today for onion. Okay, but that's what you're going to use for your wet mount. In between, a simple like rinse of your cover slip and your slide should be sufficient. If you choose to dry off your cover slip, be careful because they're really, really thin and they break really, really easy. So be careful of that. You kind of left a disaster in the back last time. I thought we were doing really well, and then on the last day I went and there was just dirty slides, cover slips still on them. I wasn't as excited about cleanup as I thought I was going to be. I actually had to do a fair amount of work to get it ready for today, so we're going to do better today. We're going to do better today. And when we're all done, we'll rinse off both the slides and cover slip, and there's just some paper towel laid out. All you need to do is just set your slide and cover slip wet, perfectly fine on those, and you can walk away, and we'll call that good for cleanup today. I'm not sure how far you'll get.
No, we're not gonna get to.
We don't know. We're doing it. You might get to Elodia. You might get to part 3 today. You might. I doubt you'll get to part 4. And I might even say we're not getting to part 4 today. We'll kind of judge that one by you. Okay. So if you have questions along the way, we are fortunate to have 24 microscopes in this classroom. So that gives you the opportunity to like have your own scope and get like, not have to be like, ooh, when's it my turn? Everyone gets to take a chance if you want. It's engaged, because we have less than 24 students, it looks like in here. So everyone gets the opportunity to scope it out.
Hey, nice.
Thank you.
If you're not sure, read the directions again.
And then
If you're still not sure, ask. That's what we're here for.